Bismillahirrahmanirrahim. This is now the paper 4 which is being continued. I've done the first 3 questions in a previous video. Now we're going to do the further questions on this video. This is the specimen for paper 4 for the syllabus 2022, which we are going to take the exam this coming season. So all the best. As I told you earlier in the previous video, in this paper 4, there is going to be no section A or section B. There's answer all the questions. So there's no section B, and this is for 2 hours, and this is for 100 marks. Now, starting with question number 4, the polymerase chain reaction PCR is used to produce large quantities of DNA from a very small original sample. The main steps of one PCR method are shown in figure 4.1. Step 1, they heat the mixture of DNA, Taq polymerase, primers and nucleotides to 95 degrees Celsius. Step 2, cool the mixture to 65 degrees Celsius. Incubate mixture at 72 degrees Celsius, and then you repeat step 1 and step 2. Basically, why do we do PCR? We do it to amplify the DNA. So we've got the original strand of DNA. Say this is the original strand of DNA. Why do we heat it? We've got to separate it. We got to separate it so that this can act as the template strand, and the new nucleotides can then bind with it. So we've actually done it to expose the bases, and so that now this DNA, we can make millions and millions of copies of it. So understand this, that why did we do it? Now let's look at the question. Explain why it is necessary to heat the mixture to 95, step 1, and then explain why primers are included in the mixture. So there were 2 different questions. One is for 2 marks and the other is for another 2 marks. So why is it necessary to heat the mixture to 95? Well, to separate the 2 strands. So yes, you separate the 2 strands, you break the hydrogen bonds between the bases, so the bases are exposed, and to produce template strand. Now part 2 says, explain why primers are included in the mixture. Now primers bind to the DNA by complementary base pairing, and they attach close to specific sections of DNA, and then DNA polymerase attaches to the double-stranded DNA. Now this word here which has been used is dsDNA, means double-stranded DNA or primers, forward and reverse primers. Now I'm going to explain this to you in a minute. Look at this diagram. I mean, this is the original DNA, right? This is the original DNA. Now we've separated it. This one goes here, this one goes here. We've added primer 1 and we've added primer 2. Now you can see it's complementary base pairing. And then what has happened is that there is going to be an extension of this. So you see this is being extended here. This is the one which has been extended here in this direction. This has been extended here in this direction. And ultimately this will be completed. So it's always in the 5-3 direction. This is completed here. So we will ultimately have 2 strands of DNA from the original 1 strand of DNA. So the primers that have been added, so we add 2 primers, primer 1 and primer 2. So it's not necessarily the same, but this is the gene, this is the place of interest which we have actually attached it. So it's very near the place of interest that we've added the primers. Now let's look at the mark scheme again. Primer binds to DNA by complementary base pairing, attaching close to specific section of DNA. Then DNA polymerase attaches to double-stranded DNA. Why? Because now the original strand and the primer causes a double-stranded effect, and then forward and reverse primers. Then coming to the part 3 of the question, explain why the enzyme Taq polymerase rather than any other type of DNA polymerase is used in PCR. Why Taq polymerase? Taq polymerase has been taken from thermophile microorganisms, and so Taq polymerase is heat stable. It does not denature at the normal temperatures which would have been taken from if you had taken it from any other animal. So it will not need to be replaced. It only needs to be replaced only after a number of cycles. Now the other polymerases you see will have to be replaced very regularly after each cycle. Here it's going to be replaced after a number of cycles, and the process is more efficient because we don't have to then worry about the DNA polymerase, and we can do it and we can get lots and lots of copies of the DNA, which is our purpose of PCR. Now let's come to the B part of the question. This is a very long question. The presence of a faulty allele of the gene BRCA2 can lead to an increased chance of developing breast cancer. There are many different faulty alleles of the gene BRCA2. So there are many, there's just one allele, but there are many faulty. So a few basis difference causes them to be different alleles. People who are considered to be at a risk of breast cancer may choose to be tested for the presence of these alleles in their genomes. A microarray can be used to test blood samples for the presence of these alleles. The microarray contains DNA probes for different faulty alleles of the BRCA2 gene. State the meaning of the term genome. Genome is all of the DNA in a person's cell, and this is combined nuclear and mitochondrial DNA. Part 2 suggests which type of cell from a blood sample is suitable for testing for the presence of these faulty alleles. Naturally, the alleles are in the DNA. So give a reason for your choice why you take the white blood cell. If you take the red blood cell, there's no nucleus in it. In fact, there's no mitochondria in it either. So and the white blood cells will contain a nucleus and will also contain the mitochondria. So we can get the DNA sample from that. Now coming to the part 3 of the question, outline how faulty alleles of the BRCA2 gene can be detected using microarray. Now, what are probes? On the microarray, we have probes. We have short lengths of single-stranded DNA. Now, what we do is these probes that we have placed on the microarray chip are actually complementary to the faulty allele. And then many copies of one type of probe is produced in each cell of the microarray. You see, the microarray has these cells, has these little pieces of glass which are called cells. So this is understand what they're meaning by the cell. Many copies of one type of probe placed in each cell of the microarray. I'm going to just explain it to you in another diagram. Sample DNA made single-stranded. So the DNA from the patient or the person we've taken, we have actually denatured it and we have made it single-stranded. So sample DNA is made single-stranded. Then we have labeled the sample DNA with some sort of fluorescent dye, green or red or whatever. Then if the person has the faulty DNA, then the faulty DNA will hybridize with the probes. That will then attach to the probes. And then the unbound DNA will be washed off. And then we will shine some UV light in it to detect if there is presence of the hybridization or not. So please understand this microarray. Single-stranded DNA, then the sample also has single-stranded DNA, then that is going to hybridize. Then this word that we have used, placed in each cell of the microarray. A microarray is a chip on which we've got these little pieces of glass which have got the single-stranded DNA attached to it. Then they ask you a very favorite question of this, because this is the ethics of genetic engineer screening. I mean, should we really do it or should we really not do it? I've discussed this in class with you all many times. Outline the advantages of screening for faulty alleles. So they've only asked the advantages. If they had said the ethics, of course, then you would have the disadvantages of it. Now, for instance, there is a family history in a family of breast cancers, and you ask this woman to have herself screened. Well, if she is negative, well, of course, that's a big relief because there's no need to worry anymore. But if she is positive, then what do we do? Well, she has to make an informed choice about making lifestyle changes. Or she has to make a choice about preventive treatment. Does she go for elective mastectomy, in which they remove the breast? So informed choices about whether preventive treatment is needed or not, then allows preventive treatment that may be cheaper than treating disease itself. I mean, if she now has elective mastectomy and later she develops cancer, what are the choices? She'll have to spend a lot of money to get it treated. Then informed choices about having children. Then positive implications for life insurance. If a woman wants life insurance, they probably want this to be tested. So these are the advantages of testing. First, I'm just going through the points again. If not present, of course, removes the worry. You're worry free then. Choice to start preventive treatment. Do you want to take the preventive treatment if it's available or not available? Then prevention can be cheaper than the disease, treating the disease itself. Then choice of whether having children or not having children, the positive implications of life insurance, and making lifestyle changes. Now you can see this is a very simple revision of microarray. What did we do with the microarray? We have sample 1 and we have sample 2, right? We have RNA, then we've labeled cDNA, so we've labeled one green and one red, and then we're mixing and hybridization, and then when we've scanned it, this is when we go to the data analysis, and the computer is going to scan it for us, whether there has been hybridization or not. So this is one example of microarray. Now, if you look at this diagram, this shows you what do I mean by the microarray cells and what do I mean by single-stranded DNA and how different sequences are immobilized on a DNA chip. Then hybridization of the floral label will be immobilized oligonucleotides. Oligonucleotides mean short strands of DNA imperfectly bound with the other forms a mispair, and then fluorescent detection of the hybrid on the chip surface, and then stringent wash of the chip. So just have a look at this, pause the video and have a look to understand the microarray story. Question 5a, the contraction of striated muscles is explained by the sliding filament model. Describe what happens in the sarcomere when the myosin head releases ADP and inorganic phosphate. So what happens to the sarcomere? Naturally, the sarcomere shortens, so Z lines move close together. So basically sarcomere shortens. If you've really understood this, you could do this only. So sarcomere shortens, or you could have said Z lines move close together. Then we come to part 2, explain the precise function of ATP in the sliding filament model. Naturally, basically that's the ATP binds to the myosin head, then hydrolyzed by ATPase, head detaches from actin, and head tilts back to the original position. So precise function of the ATP is the binding to the myosin head, then the hydrolysis of it, and then the head detaching from the actin, and the head tilts back to the original position. So this was what you had to really actually know about it, and I'm sure you all know it very well. Then we come on to during contraction, muscles use up ATP very quickly for a short period of time. ATP can be resynthesized using creatinine phosphate as shown in figure 5.1. ADP plus creatinine phosphate gives you ATP and creatinine. The creatinine formed as a result of the resynthesis of ATP is converted to creatinine. Creatinine production in the body stays fairly constant. Creatinine becomes part of the glomerular filtrate during ultrafiltration in the kidney nephrons. Ultrafiltration requires a high blood pressure in the glomerulus. Explain how this high blood pressure is achieved. Basically, how is it achieved? It's achieved because the afferent arteriole is wider than the efferent arteriole, right? So afferent wider than efferent, and that of course is the main thing that you have to understand. You see, this is the afferent arteriole, and then you have the glomerulus, and then you have the efferent. So basically this is what you had to understand, that the lumen was the afferent arteriole has a wider lumen and the efferent arteriole has a narrower lumen. Name the main filtration barrier in the nephron that allows creatinine to pass into the Bowman's capsule but stops red blood cells from passing through it. Naturally, everybody knows that that's the basement membrane, which is going to prevent it. So basement membrane. Then coming to part C, the concentration of creatinine in the blood largely depends on the glomerular filtration rate, GFR. By measuring the concentration of creatinine in the blood, the GFR can therefore be estimated. The value of the GFR can be used to assess the efficiency of the kidneys. In humans, a normal value of the GFR is 100 centimeter cube per minute. Figure 5.2 shows the relationship between GFR and the concentration of creatinine in the blood. So here we have a graph. We have the concentration of creatinine in the blood on the y-axis. Look at the graph very carefully. And we have the GFR in cm cube per minute on the x-axis. So we've got this graph. And then let's look at the question on the graph. And you can see the graph is, look at the graph in the beginning. The look at the graph is that the concentration of creatinine in the blood is high in the beginning and then is decreasing as the GFR is increasing. So it's sort of, it is decreasing and then it sort of plateaus off when after GFR of about 50 onwards, then it is sort of becoming less, is decreasing lesser. So look at it very carefully. And this graph is especially a favorite question of Cambridge. Describe the relationship shown in figure 5.2. So describe, as I tell you many times, is no biology. So describe means just a simple description of what is happening. And what is happening is the higher the creatine concentration, the lower the GFR. Then you could have said inversely proportional and inversely proportional to the GFR. So creatine concentration is inversely proportional to the GFR, and this would get you your 2 marks. Then using figure 5.2 to estimate the concentration of creatinine in the blood, that indicates a normal GFR. I worded it higher the creatine concentration, the lower the GFR. Or you could have said creatine concentration is inversely proportional, or you could have given me a data code. Data code means that you could have given me two two figures from the x-axis and two from the y-axis with the units. Now use figure 5.2 to estimate the concentration of creatinine in the blood that indicates a normal GFR. Now you know normal GFR here you can see was 100. So 100 and then you read off this value and that is 0.013. So they got you 1 mark and the units got you the second mark gram dm cube. Coming to the part 3 of the question, suggest two reasons why the GFR of a person might decrease. Now coming to part 3, as we said, suggest two reasons why the GFR of a person might decrease. Well, of course, why would it decrease if the filtration is less? You think, why? Could be kidney damage, could be cancer, could be dehydration, and if the volume decreases of the blood. And low blood pressure. Now, why would anybody have low blood pressure? And I've often told you this when I'm teaching you this in the emergency when a patient comes with a lot of crush injuries in a car accident. He has lost a lot of blood, maybe internal bleeding, maybe external bleeding, and that is the time when his kidneys are going to go into a shutdown phase. And the first thing we do is we restore the blood volume by putting up a drip. So the blood pressure can drop if there is massive internal bleeding, or even if there is fracture of a bone or something, and there's internal damage. So there's a lot of blood loss. You cannot see the blood loss, but maybe it's internal blood loss. And that results in the volume decreases, and that of course results in low blood pressure. So please remember that in any question that might come in the future exam. Now coming to question number 6, the black pigment melanin contributes to hair, skin and eye color. Melanin is produced by cells known as melanocytes. Tyrosinase is an enzyme involved in the production of melanin. A study was carried out to investigate the effect of an extract of starfish, whatever, on the activity of tyrosinase. So we took an extract from starfish and we wanted to study its effect on the activity of tyrosinase. Tyrosinase is an enzyme. So either it's going to increase the rate of enzyme activity or it's going to decrease the rate of enzyme activity. If it's going to decrease, that's going to be an inhibitor. If it's going to be increased, then maybe it's some sort of a cofactor or something. Table 6.1 shows the results of this study. So look at the concentration of extract. So we had 0 extract, so it was 100 was the activity. Then we added 4 micrograms per cm cube of the extract and it was 90. And then we added 8 and it was 77. And then we added 16 micrograms and it was 68. And then we added 32, so it was decreasing. Now you can see it's decreasing right throughout. 56, 46, 32. So as the extract is increasing, the activity is also decreasing. So the extract must have contained something which is reducing the activity of the enzyme. Now what could that be? That could only be an inhibitor. Suggest how the starfish extract affects the activity. Suggest means it could be any answer. So the greater the concentration of extract, the lower the activity of tyrosinase. Simple, you got it from the table. Nothing else to know from previous studies or anything. So the extract acts as an inhibitor. That's the second mark scheme point. Now this inhibitor probably binds to the active site or to another allosteric site. Maybe it could be that. Maybe it alters the pH and or the extract can change the active site or it can denature the active site. So all these possible answers were allowed, and but from this table you have to guess all that. Just worded it for you. Greater concentration lowers the activity. Greater concentration of the extract lowers the activity. Extract acts as an inhibitor, binds to active site or maybe binds to the allosteric site. It maybe alters the pH, and this extract changes the active site. And then coming to the B part, the dominant allele of the TYR gene codes for the enzyme tyrosinase. In people with albinism, the melanocytes do not produce melanin. This is caused by a recessive allele of the TYR gene. So small r would be the recessive gene, big R would be the TYR gene. Explain what is meant by the term recessive and allele. A recessive means that which is not expressed in the heterozygous state. So there is a small r, but is not expressed in the heterozygous state. Allele means different varieties of the gene. We have big R and we have small r. So different varieties or different forms of the gene. Then coming to part 3, construct a genetic diagram to show how a man and a woman who both produce melanin could have a child with albinism. Use appropriate symbols to answer and state what they represent. Basically, A, you can say, is the normal pigment, and small a is the no melanin or albinism. So the parents have to be heterozygous. Why? Because they were normal. They didn't have the disease, so they had to be heterozygous. And that I always tell you is that you've got to figure it out what our albinism going to be. It's going to be small a, small a. So one A has to come from the father and one A has to come from the mother. So it's a very simple way of really deducing that. You cannot have a child who could have been big A, big A. If one parent even had big A, it's not possible that if one parent is big A and one parent is big A, small a, and then the child can be small a, small a. That's not possible. So one small a has to come from the mother and one small a has to come from the father. So that is how the person will have albinism. Question number 7. Figure 7.1 shows a red deer, Cervus elaphus. Red deer feed on a wide range of plants. The number of red deers in the UK increased from 135,000 in 1960 to 360,000 in 2010. Environmental factors affect the population size so that numbers do not continue to increase. Suggest environmental factors that may prevent further increase in the size of red deer population. Population is, like we say, the population of Pakistan is so and so many numbers, is the number of one species at a particular time and place. So basically, it must have been competition for food, disease, predation, loss of habitat, pesticides, maybe there's some change in the climate. So all these factors would have kept the population constant. Just mention it: predation, competition for food, disease, loss of habitat or breeding sites, so their numbers are remaining the same. Pesticides and herbicides would destroy the plants which they eat, and change in the climate, less rain, so less grass or less plants, so that could also affect the number of population would remain the same. The body mass of red deer shows wide variations within a population. This is shown in figure 7.2. We have the body mass, we have low body mass, and we have some in the low body mass, and then we have some in the, so mostly we have here, and this is the largest number, is this is the medium body mass and the high body mass. So just look at the graph carefully, try to understand it. It's the mass of red deer shows wide variation within a population. Then part B, a selection pressure. A selection pressure acted against the low, acted consistently over many years against red deer of low body mass. Against, consistently against the low body mass in a population. Sketch a curve on which to show the pattern. So of course the curve would of course shift to the right, because the low ones are going to be killed. So sketch a curve to show the pattern of variation of body mass in this red deer population after this time. There's only one mark for that. So you should have taken this and you should have drawn it in this way. So it goes to the right. Name the type of force of natural selection that is acting on this population. Naturally, it is directional selection. It is moved to one direction, to one side. So directional selection, that would get you a mark for that. A selection pressure acted consistently over many years against red deer of medium body mass. So acted consistently over many years against red deer of medium. So the medium is somewhere here. Sketch a curve on figure 7.4 to show the pattern of variation of the body mass in this red deer population after this time. So they're going to be same position, but they're going to be two peaks, right? And then of course they've asked you to name this, and this is called disruptive selection. So basically it's something which much in the books and in your syllabus as well, so you've done this many times. So this would be called disruptive selection. So in this video we've covered question 4, 5, 6, and we've also question 7, and the rest of the 3 questions, which are 8, 9 and 10, will be done in the next video. And thank you very much, and please do subscribe.